Welcome all to the AI seminar series. Today we have Dr. Yufei Huang from UTSA joining us to share his research on intelligent biomedical research. It's my pleasure to introduce Dr. Huang, who has received his PhD in electrical engineering from SUNY Stony Brook in 2001. And since then, he has been a faculty member at UTSA. His affiliation is with Electrical and Computer Engineering Department at UTSA, but he also has an appointment with the University of Texas Health San Antonio in the Department of Population Health Science. His expertise is in the areas of computational biology, brain-computer interface, machine learning, and AI. He brings in a unique combination of these thrusts, whereas his current focus is to study the functions of mRNA using machine learning and high throughput sequencing technologies. And his lab has developed several widely used M6A data analysis pipelines. And Dr. Huang's lab also uses AI systems for precision medicine and passive EEG-based brain-machine interaction systems for understanding human cognitive behaviors. For his work, Dr. Huang has received several awards, but to name a few, he has received the National Science Foundation Career Award, the UTSA Presidential Achievement Award on Research Excellence, and several Best Paper awards. He sits on as an associate editor for several journals in signal processing and biomedical research. And a special tidbit here, I was Dr. Huang's student as a first-year PhD grad, and his probability and statistics class was really foundational in my understanding of these learning systems. So thank you, Dr. Huang, for joining us today and sharing your research.
Thank you, Derisha. Thanks a lot for organizing this. If you don't remind me, I didn't really remember that you were in the class, but it's great to know you. So thanks a lot for organizing this. I think it's a great opportunity for me to present to the community some of the research that we've been doing over the years on applying AI for biomedical research. So let me start by really tell you a little bit about the so-called recent hype in biomedical research. I believe most of you now aware of this AI hype in mostly computer vision, natural language processing, but probably has little knowledge about AI applications in biomedical research. But I just want to tell you that there's a equally hype in biomedical research in applying AI. So there's one of the evidence is that there's a significant increase in the number of publications in this high-profile venues like Science, Nature about AI, especially deep neural networks applications in, for example, functional prediction of non-coding sequences, variable calling, single-cell analysis in radiology, pathology, dermatology, for predicting clinical outcomes, drug selection and design, and improving healthcare in general. So as you can see, all those papers are published in either Nature or Science, and I can give you more and more this type of examples. Here's a so-called non-exhaustive list of potential applications of AI in biomedical and biomedicine research. So now in medicine, AI has been envisioned to really function as a virtual medical coach to provide individualized health recommendations across human lifespan. So how can AI really help biomedical research so that we can materialize this vision? So I want to share with you some of my thoughts. So I believe integration of AI and biomedical research will really usher us into a new research area. I call this intelligent biomedical research. So where in this research, AI functions as an intelligent assistant to really work together with experts like doctors, or biologists to accelerate the research cycle of experimental design, data analysis, and hypothesis generation. So this is a typical cycle of biomedical research. So to achieve this, we need to really equip such an AI agents with the so-called intelligence, like a human, to be able to communicate, to learn, and also learn fast, to predict, to understand, to reason, and finally, to be creative. Okay. So despite this recent hype, and despite this ubiquitous use of the word AI, so the current so-called AI algorithm at the best can learn, but not fast yet, and predict to some level. And there's really a little sign of current AI model to possess other intelligence I just described there. So to be able to really perform the functions of this AI agent that I envisioned. So why suddenly there's a hype in AI? So I contribute this recent hype to two key factors. The first factor is really because of the so-called deep learning revolution that everybody is aware of. Right? So this deep learning jumped into public attention due to its ability to provide human level and in many cases superhuman performance. Initially, computer vision and speech recognition, and later in almost any subject area that you can think of, including, for example, image generation, video game playing, this chess play AI, and currently natural language processing is a very big area where AI made significant advances. Okay. So, and the second factor is data. So we all know that the deep learning is really data hungry. So all previous extraordinary performances of deep learning have been achieved by training on huge amount of data samples. Okay. So in biomedical area, advances in high throughput biomedical equipment, such as high throughput sequencing and ubiquitous use of health-related wearable devices, the concerted effort by hospitals and federal agencies to collect electronic health records, genomics, brain imaging, and mobile health data has really provided a fertile ground for us to apply deep learning. So now, so I hope now that I convinced you that this is really a good time for us to apply, to really think about developing AI for advancing biomedical research. However, there are big challenges that still prevent us from achieving what I envisioned. So I listed some of the missing parts here. So one of the key missing elements is this theoretical framework for building AI systems. Okay, so namely, how human really makes decision, how human really reasoning. So there's really little knowledge to really understand how human makes such decisions. So in addition to this, if you look at the current so-called deep learning models, most of them actually use little prior knowledge, but that's in contrast to how human really makes decisions. We use a significant amount of prior knowledge in making decisions. And also these prior knowledge are organized in a causal fashion. So these are not current deep learning model possesses. So in addition to that, the current deep learning model is very much a black box. So this, where human is highly intelligent, we make predictions and also enable and also be able to really reason why we make such decisions. So this is also what the current deep learning model is lack of. So in addition to this, I mentioned about being able to reason, to create. So this is the current deep learning model is nowhere near being able to really reason intelligently, and let alone create new knowledge and new understandings. Okay. So another challenge is so-called small samples. Actually, human don't learn using a large amount of samples. That's what deep learning does. So human learns using a very small amount of samples. So how we can equip deep learning to learn new knowledges and with small samples is really a tremendous challenge, but it's highly relevant to be able to create intelligent systems. So other related areas, including such as security and ethics. So human is a moral entity. We possess, this um, empathy and a moral understanding towards anything that we encounter. But a deep learning model has no signs of the current deep learning models, no signs of such moral standards and or ethical understanding towards anything. So that's another level of research that's all actually being investigated by this community too. But these are all the things that, in my opinion, are important in order for us to build a AI system to be able to achieve what I envision to achieve. So I just want to bring your attention to this so-called the human cognitive systems. And there's a very nice book written by Daniel Kahneman, who is a Nobel laureate. So he described this two systems of human cognitive systems. So this is a system that now are in the mainstream of deep learning research for people to build AI systems. However, there's still many missing parts in this understanding, in this system, capturing human cognition. So now, before I move on into my own research, I want to quickly provide an overview of deep learning models. So I believe most of the people in this audience are very much familiar with deep learning, but I still want to quickly go through this. So what I want to tell everybody is that one of the reasons why deep learning is so popular in recent years, when compared with traditional machine learning model, in addition, so the reason, in addition to deep learning model doesn't require extracting features, another key reason is the construction of deep learning model is very modular. So I listed a few modules where people use to construct deep learning models. So they are dense layers, convolution layers, residual layers, graph convolution layers, recurrent layers, attention layers, prediction layers, so on and so forth. So you can pick and choose, combine them together, and to really build deep learning models. So as everybody know in this audience, so the current deep learning models are normally categorized into two areas, two fields. One is called supervised deep learning models. So I'm showing you some of the most popular supervised deep learning models. I just want to point out that these models, for example, deep neural network or DNN, CNN, convolution neural network, graph CNN, and so on and so forth. As you can see, they are all modular structures, constructions by using these modules. Okay, so supervised deep learning model functions to really predict, taking some of the data. So that's an example of a CNN. So supervised deep learning model takes the data as an input and predict the so-called outputs or the labels of the data, right? To be able to train such a supervised deep learning model, we need to provide supervisions to the model, namely provide so-called training data. So in this case, we need to tell the model, give the model a pair of so-called ground truth input and outputs. So for example, an image of bird and the label bird. And we need to provide the model not just one pair of such samples. We need to provide the model with lots of these such samples. In many cases, millions of these such samples, and then use pretty intelligent training algorithms. In the case of deep learning, this is another reason why deep learning is so popular. This is because training is also fairly standardized. So this dominant algorithm that used to train deep learning model is the stochastic gradient descent algorithm, right? And powered by backpropagation. So using such algorithm, you can train deep learning models, or in this case, CNN models. So when I say train, it really means to estimate the model parameters. So after you train, and you can take that model and apply to new images to be able to predict new labels. So this is what a supervised deep learning model does. So the second category of deep learning models is the unsupervised deep learning models. Okay, so the goal of unsupervised deep learning model is to really understand images or understand data. Okay, so in this case, the data includes only the input samples to the deep learning model, images, for example, and it doesn't have so-called labels. So it's a pretty tough question as to what exactly we're learning in this case, right? So, one of the things that we're learning is to learn the so-called representations. So such that, I can quickly jump to this page, such that once you learn, you're able to, for example, the input are these images or handwritten digits. You're able to understand the structure of data to be able to know, in this case, that there are 10 different groups of samples. So how do we achieve this with deep learning? So one of the most popular models is called autoencoder. So the autoencoder, I give an example of autoencoder here. It takes, it's a deep learning model, so you can use these different modules to really build them. So for the input, so this autoencoder takes a data, takes a data sample as an input, and spits out the so-called code. Code is considered as a low-dimensional representation of the data. Okay, so the way how you train autoencoder is that you add a so-called decoder on top of the encoder, on top of the code, to really reconstruct the input. And you train the model such that the reconstructed input is as close to the original input as possible. So once you train the model, you can throw away the decoder. Of course, decoder is still useful, but just for the sake of understanding the code, you can throw away the decoder. And now you can take the code and, for example, in this case, you take a two-dimensional code, you can visualize the data, which originally sits in thousands of dimensions. And now if the deep learning model this autoencoder is trained well, now using the code, we're able to really to understand the categories, different classes of the data, which will be very difficult to understand if we use the original data dimension. Okay? So in addition to that, so you can also take the decoder to be able to generate samples. You can manipulate the code and to be able to, for example, generate new samples. So so besides autoencoder, another line of very popular unsupervised deep learning model is called generative adversarial networks. So generative adversarial networks, the goal is still the same as autoencoder. Well, I should back up one step. So I should correct myself. The goal actually is not exactly the same as autoencoder, but the goal here is to train a so-called generator to be able to generate new samples from some of the code, right? So, or in other words, the goal here is to train a generator to learn the distributions of the data. So data can be images. Okay. So I'll give you some of the examples of GAN. GAN is really one of the revolutionized inventions in deep learning area. So here are some of the images generated by GAN. So what I want to tell you is these are images of people that do not exist on Earth. So they are purely synthesized images. But look how realistic these images are. So it really underscored the power of GAN. Okay, so GAN actually, so there's a huge there's a huge activities, good or bad, beneath using the GAN. So here are some examples. Deepfake, this website Deepfake, it's really, really popular that actually generates fake images, fake articles, and so on and so forth. And the engine behind deepfake is GAN. Okay, so here's a picture that produced by GAN. So this image actually is auctioned for almost $500,000. Okay. So GAN is also being used in biomedical research to really design new drugs. So there's a huge application of GAN. So I believe most of people also realize. So another, well, this is not exactly a GAN model. So this is the so-called self-attention model. So self-attention model is the current state of art natural language model. It's not exactly a unsupervised learning model, but it's often being used as an unsupervised model to learn languages, to learn text specifically. So I'm showing you an example of this article generated by the state of art self-attention model called GPT-3, produced by OpenAI, trained on 50 billion these words. So you can read this article that generated by GPT-3, and I don't think anybody can tell this is a machine-generated article. Okay. So with all this background, let me jump now, switch gear. Let me see how much time I have. Okay, good. So let me switch gear to really talk about my own research. And so I have two lines of research as described by, introduced by Darisha at the very beginning. So one focus of my research is on understand human cognition using specifically EEG data. So EEG measures electric signals as a result of human brain activities. So the goal of that line of research is to really sense the cognitive conditions using EEG and be able to also predict mental states, and then to see how we can use this knowledge to be able to improve human performance and health. So another big focus of my research is on genomics, specifically at the level of DNA, RNA, and genes. So we try to really apply AI and to understand the regulations, interactions among all these molecules, and to be able to elucidate the molecular functions and then translate that knowledge into medicine. Okay, so the basis of my research or the tools that we use are very much machine learning, deep learning, and now AI. So let me quickly run through my research on brain-computer interface, even though today's focus of my talk is really on genomics. So we're one of the member of Armory Research Lab flagship this consortium called Cognition and Neuroeconomics, CTA. So my lab actually is charged to build deep learning models to be able to predict these different cognitive behaviors from EEG. So we did build models, deep learning models that takes EEG and to predict visual attention, human decision, and driving fatigue. So one of our recent work actually is to use GAN to be able to learn from existing EEG data to generate new EEG data that mimics this EEG, mimics the EEG data that we sampled, that we collected, that measure human cognition. So if you're interested, you can go to this paper and read more, or talk to me about some of the details here. As I said, the focus on today's talk is really on our deep learning work for genomics. Okay, so to start, let me provide some background on genomics. So I want to bring your attention to what I'm talking about is really cell. So I'm looking at I'm showing you this the cell mammalian cells that includes a nucleus inside the cell. So I want to bring your attention insight into the nucleus. So what I'm really talking about today is DNA. So the DNA is inside the nucleus, and DNA itself are two chains of nucleotides. So in this DNA, there are 4 different types of nucleotides. They are guanine, cytosine, adenosine, and thymine. So, and we normally use the first letter to denote these 4 different nucleotides, or we call them a G, C, A, T. So DNA, one of the chains of the DNA, you can consider that as a long, it's a long sequence of these 4 nucleotides. So we also call these nucleotides as a base pairs. Okay, so in human DNA, so this long sequence includes 3 billion these letters, ATCG letters. Okay. However, in DNA consists of 2 chains, and these 2 chains actually pair together through the so-called base pairing. So base pairing refers to a specific pairing between these nucleotides, and specifically thymine always paired together with guanine, or C always paired with G, and adenine always paired with thymine, or A always paired with T. And then because of this pairing, this pairing also enables 2 chains to really tie together. And also because of this pairing, if you know the sequence in one chain, you also know the sequence of the other chain. Or in other words, these 2 chains have exactly the sequence, but in a so-called complementary fashion. So for example, in this case, if you know the order of the sequence in one chain is A, G, T, A, C, G, you know exactly the sequence in the other chain through the so-called base pairing. Or if you know this is G, then so you know the nucleotide in the other sequence will be C, or G, or T, or A, and C, T. Okay. So why DNA is so important? So DNA actually stores the so-called genetic information. So that genetic information is coded as this ordered sequence in this long DNA chain. Specifically, this ordered sequence functions very much like languages, like words. So a specific combination of specific order of this letters actually carries the genetic information. For example, this GC, AT, GA could carry some certain genetic information. So what's the importance of such a genetic information coded in the sequences? So that sequence, this genetic information actually dictates the production of proteins. Okay, so a specific sequence would lead to a specific production of a type of a protein. Okay, so in human, so there are about 20 or 25 segments in the human DNAs that functions to store genetic information. And these segments in DNAs are referred to as genes. So therefore, in human, we have about 20 or 25 different types of genes. Okay? So each gene will function to produce 1 or more different proteins. Okay? So because protein is the basic building block of any human beings, so the change in the genetic information will fundamentally change the protein and therefore the phenotypes. So and the phenotypes, our appearance or disease are all the cause are all the result of the phenotypes, and therefore the change of the code. So here I'm describing to you the process of going from this genetic code stored in DNA to the final product of the protein. So this process involves 2 stages, namely transcription and translation. And these 2 stages are described as central dogma of molecular biology. So the first step, transcription, concerns transcribing the code stored in DNA into an intermediate molecule called mRNA. So mRNA is a short for messenger RNA. It's an RNA. So the difference between RNA and DNA is that, so first of all, RNA is also a long chain of nucleotides, but it's a single chain of nucleotides opposed to double chain nucleotides. But the only difference in terms of this composition of nucleotides between mRNA or an RNA and the DNA is that it consists of also 4 letters, but they're not ATCG, they are AUCG, or that U, uracil, replaces thymine in the case of the DNA. But regardless, it's just mRNA is also a long chain of these nucleotides. Okay? So and the second step in this central dogma takes the mRNA, which is a copy of the code in DNA, and then bring it for the production of protein. So I give you a more give you a depiction of how this 2 stages really happen inside the cell. So what you're looking at here, this is the nucleus, and the DNA actually sits in the nucleus. The reason why it takes up 2 steps is because of the protein production factory known as a ribosome sits outside the nucleus. So to be able to really take that code stored in DNA into the ribosome to produce protein, so we need a messenger, which is this messenger RNA, so that started inside the nucleus and that copies the codes stored in DNA through this process called transcription, and then bring the codes outside of the nucleus into the ribosome, and then dictates the production of the proteins here. So the second step is called translation. Okay, so you might, so this describes the process of translate the code stored in DNA into protein. But you might ask, so for humans, our genetic information is very, very similar. So, and then you might ask why we're so different. We're still so different. Every individuals are virtually different. So the reason why, even though the genetic codes between us are very similar, but on appearance we're very different, is really because these 2 processes, transcription, translation, is under tight control, and that so I'm showing you at each stages some of the words that describes these different type of controls, which probably don't mean too much to you, but I just want to tell you that there's a very sophisticated control of these 2 stages. And the reason why we're different is really because this underlying control of transcription and translation is different. Okay? And this translation and transcription, this control of translation transcription is also a time series process. It changes all the time. Okay? So this type of control involves, for example, proteins and sometimes protein binding into the DNA and protein binding onto the RNA as well. And it possible also involves modifications of the nucleotides inside DNA and in mRNA, modifications through chemical modification or mutations. Okay? So what I want to talk about today is how we can use, however, this type of control. Now it's the center of the biomedical research, but there's still many elusive questions to be answered. So what I want to show you today is how we actually use AI to help us understand the control of these transcription and translation processes. Okay. So I want to talk about the few examples. The first example I want to talk about is predicting the transcription factor binding in DNA. So what I'm talking about is this binding of a protein. a type of proteins referred to as transcription factor. The transcription factor binds on DNA right in front of a gene that stores genetic information. Such binding actually functions as a switch to really control the production of mRNA. So it turns on and off in a very simple fashion the production of mRNA. So knowing which transcription factor binds in front of certain genes is very important for us to understand the regulation of transcription. So one of the questions actually we're looking at is to be able to predict which protein or which transcription factor binds by simply looking at the nucleotide contents of DNA sequences, for example, in front of the gene. So this question can be simply described by this cartoon where you're building a deep learning model. The input to the deep learning model is the sequence of nucleotides, sequence of letters, ATCG. For example, 100 letters, we call 100 base pair long, and we asked whether this transcription factor CTCF binds in this sequence or not. So the sequence is taken, for example, from the front of this gene. Okay, so there's a seminal work actually published in Nature Biotechnology in 2015, and this work actually is considered as the first work that applies deep learning in genomics. So which actually designed a deep learning model that specifically answers such question. So this deep learning model is a CNN model, and this model actually this work built over 1,000 CNN models to predict the binding of a large different variety of transcription factors. So one of the issues with even though this model can predict whether a transcription factor binds in the sequence or not, it cannot predict exactly in the sequence the transcription factor binds. Okay. So we take on this challenge and then ask whether we can build a model to be able to precisely predict binding sites in this 100 base pair long input sequences. Okay. So essentially, we want to build a deep learning model, okay, so that takes the sequence as an input and outputs the locations. So the locations of the binding is described as a vector with the same length as input that includes 0 and 1s as the elements, where 1s indicates the location of binding. So that's an example of the outputs. So the question here is, how do we build a model like this? Okay? So clearly, this is not a classification problem anymore. So because the output is not simply 0 and 1, and notice that the output is of the same dimension as the input. Okay, so this actually motivates us to really look into computer vision and see whether there are words that also produce outputs that have the same length of the input. So here is one such work in computer vision. It's referred to as segmentation. So the goal is exactly the same, where the goal of such a segmentation model takes the input of image and outputs generates an outputs of the same dimension as the input with 0 and 1s, where 1s indicates where, for example, the dog is. Okay, so and such model, one of such model is called a deconvolution model. It takes a convolution model that outputs yes or no, and then flip it and takes that information of yes or no and blows it back into an image. Okay, but the image contains only 0 and 1s. So such model is called a deconvolution model. So we take inspiration of this deconvolution model and take the steep binds produced in this Nature Biotechnology paper, and then flip it, okay, and take this train on the ground truth input binding site and the ground truth locations of the binding sites. Okay, so that's that's very much how we build such model. Okay, so we just flip it and to construct a deconvolution model, and so we train this model using about 60,000 ground truth binding sites, so the results actually show that our model provides significant improvement. If you look at the IOU and the F1 score over one of the existing approach that is not deep learning based. So this is the first deep learning model that enables prediction of the sites, okay, precise location of the sites. And so our prediction actually also confirms some of the known knowledges for the CTCF binding, which describes the significant region of the binding starts from the 10th and or in between 10th and 11th base pair at the center of the binding site. So this is exactly how we predict it. Okay. So and also our model is able to confirm the so-called binding motifs of this transcription factor. So it's a comparison of the known motifs up here and the predicted motifs by our model. So you can see significant consistency. So now let me.
Yeah. Sorry. I think this might have been answered, but there's a question in the chat asking what is the significance of predicting this binding?
What's the significance of predicting the binding? Okay, so one of the key questions in this genomics is to know how a gene is regulated such that it produces these either proteins or mRNAs. Okay, so to understand how gene is regulated, a key question is to know which transcription factor regulates that gene and when that transcription factor regulates. So to know which and when transcription factor regulates, we need to really understand where that transcription factor binds and how transcription factor binds. So our model actually provides that ability to predict where transcription factor binds and which transcription factor binds in the DNA sequences using the DNA sequences. I'm not sure whether that answered your question.
Yeah, they said thanks, so yeah, thank you.
Yeah, so you can always we can always discuss offline if you have additional questions. Right? So the key focus here for all the work I'm talking about is to understand the regulation of transcription and possibly translation as well. So the next topics I want to talk about is another type of regulation of transcription through the so-called modifications of the nucleotides, but now inside mRNA. So I talked about the binding of a transcription factor into DNA. Now I want to switch our attention to look at mRNA. And just to tell you, mRNA itself can be regulated after it's been transcribed from the DNA. So the specific regulation is called modification. Specifically, it's called M6A. So this is a modification I want to just show you here. It's a chemical modification of adenosine in mRNA. So by adding a so-called methyl group. Okay. So this chemical modification of the structures of the adenosine can change the functions of mRNA. Therefore, the regulations of mRNA. So that the change of the regulation of mRNA could result in different number of mRNA being produced. As a result, therefore, different amount of proteins being produced. So that can lead to eventually changes in the so-called phenotypes. So that can very much be the cause of developing cancer versus normal. Okay, so so this is a very recent discovery of these modifications in mRNA. So I'm just showing you some of the stem work that enables this discovery. So and also this modification is widespread in mRNAs in about 25% of different type of mRNAs. So it's just to show you the significance of such modification. And because of this widespread of modification mRNA, so it's inevitably it will impact the functions and regulations of mRNA. So here I'm talking about one type of mRNA modifications are called M6A, but I just want to tell you there are really many different types of modifications in not only adenosine but also cytosine, for example, in different modifications, okay, in mRNA. So this is one of the really very active area of research. As Durisha also introduced in introduction, we're one of the leading labs that developing analysis tools to really understand, to predict where these modifications are and the functions of modification. These are some of the tools that we developed in the lab, and some of them are widely used in this community here. So what I want to tell you, let me skip this. What I want to tell you are two stories. First of all, this is very much like in the case of this predicting transcription factor binding sites. Here, we're looking at a sequence of mRNA, and we want to predict whether there's a modification. Okay, for example, we take the sequence as an input, we focus on A. In the middle of the sequence, we ask whether this A is modified, or whether there's a potential for A to be modified. Okay, so and you can ask this question for other modifications. So you can ask whether this A is modified in this particular fashion called M6A, or it is being modified in another fashion called M1A. So this 1 and 6 describes the location of modifications. Or you can take the sequence, you pick and C, you ask whether C has a potential to be modified. The reason you can ask such question is because the modification, to enable that this nucleotide to be modified, there needs to be protein, RNA binding protein involved to really add modification onto these letters. So this binding of RNA binding proteins leaves footprints in the mRNA. So we actually want to use deep learning to be able to uncover those footprints in mRNA that are associated with different bindings. Okay, so that's very much the goal of this work, or in other words, to build deep learning models to be able to predict, given the sequence, whether there are potentials for these nucleotides to be modified. So this is clearly a supervised learning model. So we need to have ground truths. So one of the sources to get these ground truths are some of these databases. So this one example, one database that catalog these type of modifications. So what I'm showing you here is for these different modifications, I'm showing you 5 different modifications. These are the number of known modifications that are cataloged in these databases. Okay, so look at this one particular case, M7G. So there are only 30 samples. Or in other words, you have to build a model using 30 samples or 200 samples. It's extremely challenging, almost unimaginable with deep learning. So one of the key challenges here is how do we build deep learning models to be able to handle small training samples? And also there's a highly imbalance in the data. So namely, the so-called negative samples or the sequences that don't contain modifications are vast because most of mRNA regions don't contain modifications. So there's a highly imbalance between positive and negative samples. Okay. So how do we really address these issues? So one of the strategies that we take here is based on this hypothesis where that sequences that mRNA sequences that are not covered by the training samples, by the small training samples, should also contain modifications. The reason why there are only small number of catalog samples is really because of limitation of technology. or in other words, we haven't really been able to discover these modifications yet. So so in a sense, there are many hidden gems in the sequences that are not being discovered. So our question is whether we can use deep learning models to be able to uncover these hidden gems or hidden regions, right, which carries the footprints of the modification. Okay? So that strategy that we use is to use a so-called unsupervised learning model to first take, without really taking these training samples, we take the entire mRNA, a very long sequence of mRNA, and to scan through the entire mRNA to really learn common patterns. Okay, and some of the patterns are modification footprints. Okay, so this is the first step: train an unsupervised learning model to learn these patterns. So so very much we're deploying this unsupervised learning model that I described before, and specifically in this case, we're using GAN to learn these patterns in the mRNA. Then afterwards, we take so that specific model that trains through after this unsupervised learning process is called feature extractor. So that takes essentially a sequence as an input and convert it into the code. So the code is a better representation of the patterns. Okay. So once we train this feature extractor, and then we can apply the feature extractor to the limited code, the limited ground truth samples. Okay. So the ground truth samples, because it's trained to learn patterns. Okay. So the feature extractors will be able to really extract useful patterns from these limited samples, which otherwise will be very difficult to learn using limited samples, and then build a classifier on the code. So that's very much the strategy that we take. Okay, so that's first of all, this is the so-called GAN model. It's not a traditional GAN model. It's a so-called ALI. So it's an adversarial learn inference model. So this model not only enables us to generate new sequences, it also enables us to learn the code, which cannot in the traditional GAN model. So we trained this model in about 40 million samples, mRNA sequences. And it took about 4 days for us to really train such a model. Okay, so once we train the model, we take this encoder, which is one part of this Ali model, and then take the encoder to the training sample, to the limited training sample, and convert the training sample into the code. So the code is a better representation of the underlying patterns, and therefore the modification footprints. And then we train the classifier. We actually train a simple classifier, SVM. We did not really train deep learning, once again, because of the limited samples. We train a simple SVM on the code. And here, what I'm showing you is the improvement of the performances across 8 different modifications. Okay, and for M6A, where we have significant number of samples, and we can compare with existing models. One of the state-of-the-art models, as you can see the improvement of the performance in terms of the error under the precision recall curve. Okay, so and also using the model, we're able to uncover the so-called the footprints in terms of the motifs. So these are the sequence motifs that describes the so-called footprints of the modification, and for all these different modifications, and many of them right now there's a really lack of knowledge for the footprints for many of them. So this is almost the first time that we present the footprint for this variety of modifications in mRNA. So let me see the time. Oh, well, I guess my time is really up. So let me just stop here, or take the last 2 minutes to quickly tell you that so in addition to taking sequences, what I talked about are really most of the work using applied deep learning in understanding functional binding, functional modification, and sequences. In addition to that, you can look at the output of mRNA and then predict for example, you look at the mRNA, output of mRNA is referred to as a gene expression. And so it becomes a routine now to really collect gene expression from patients. Okay? And one of the goals is to take the gene expression to be able to use it as a surrogate for example, for diagnosis, for predicting the cancer types, for predicting patient survival, for screening drugs. So there's one line of work that we did to predict cancer types, primary cancer types, and predict the state of metastatic cancer types from using the gene expression data and using the so-called one-shot learning to be able to really predict cancer types using only one samples, as opposed to using a large number of samples. Okay, so I'll skip this part of the talk and just to conclude. So I hope I convinced you today that deep learning has really reshaped the landscape of biomedical research. So I talked about a few examples that we conducted in the lab to show how deep learning can help us to improve the knowledge of the genomic functions. However, we're really far from achieving the true AI. But this is really an exciting time to really work on AI biomedical research. So here are some of the challenges, some of the topics that I think we can focus on, including, for example, building. So most of these deep learning models that we use are computer vision based deep learning models. They're not really designed to really model biology. So there's an urgent need to build deep learning models that fits into the need of these biomedical data. So also, most of these deep learning models are lack of interpretability. But in biology, knowing why, it's the central question. So building deep learning model to be able to interpret, to be able to really explain the underlying biological functions in addition to this prediction is essential. And also to build deep learning model to be able to reason what is a better, for example, what's the better practice in selecting drugs after you train the model on a variety of the data is also essential. So and other relevant questions, including especially when we move towards this clinical research, includes security. So how secure this deep learning model you build, even though it can achieve human level or superhuman performance, but how secure this deep learning model is, and whether one can really trust the result that deep learning model produce are also central questions in the biomedical research. So let me finally conclude by thanking my collaborators and funding sources for supporting this research. So I'm happy to take questions.
Sure, there's one question in the chat. Tej, do you want to ask the question?
Yeah, sure. Can I ask? Yeah. Well, the thing is, you've gone through several models. Actually, the talk was really interesting. Thank you. And I got to see different applications. And you showed us so many different deep network models for, like, extracting information and predicting different results for different types. For example, towards the end, you even showed us cancer. And it's very effective. But the thing is, can you do the models have any sort of method for explaining why they make a particular inference decision? And how do we, it kind of is related to the interpretation that you spoke of at the end, that last point.
Yeah, this is really hit right on the point, right? So what I can tell you is most of this, first of all, whether there are approaches that enable us to interpret, yes. So first of all, most of these models are CNNs, as you see probably in my presentation. So there is a line of research that builds models to be able to interpret CNN models. So most of the tools that we use in this domain for biomedical research are also based on those tools that develop for computer vision for understanding CNN models built for computer vision. So the question here is how effective these interpretation tools are. One of example, for example, deep lift, right? How effective they are. So what I can tell you is they're not very effective. So the key reason, I can give you some of the reasons why they're not effective. So for example, in genomics, when it comes to understanding of a cancer, okay. So oftentimes, so in this field of research, if you talk to biologists, they keep on asking you what are the key genes or marker genes. So the reason why they're not asking, what's the global regulation of these genes. Rather, they're interested in a few genes. There's a biological reason behind this. It's really because, for some of these functions, cancer, for example, there are only a few genes that dictates the course of this cancer development. So therefore, to be able to interpret that algorithm, that models should be able to pinpoint a few inputs, a few elements of inputs that are important for the prediction. But this is not the current this interpretation model is designed for. So yeah, just in short, they're not really effective. So there's this is really one of the key area of research in this domain.
Okay, cool. Thank you. Thank you so much.
I know we are past time, but I had a quick question. Maybe we can have a detailed answer to that later. But in both your EEG signal generation and using GANs and also in your mRNA, how are you validating that the new signals that are being generated are valid or are they in the right form? So is there a process you're using for that validation?
Yeah, so it's a great question. It's a great question. So yeah, you can read our paper. It's really a challenging question. So what we did there is so we focused on a specific set of data where we have lots of prior knowledge as to what's the shape of the EEG data, where the signal comes from. So we have that prior knowledge. So we use that prior knowledge to really gauge the effectiveness of the generated samples. So in addition to that, we did come up with some of the quantitative approaches to really measure how similar the generated samples are to the real samples. So one of the approaches that we come up with is to measure the distances between the distributions of the real sample and the generated samples. So you can read the paper that we come up with, but overall it's a very challenging question. So yeah, so I think we need more research in that direction.
Thank you. I think one last question. Sorry, we are a little over time, but I think I want to make sure everyone's questions are answered. Are there any established measures or standards considering how a model's prediction confidence factor can be considered?
Can you repeat the question again?
I think, Anurag, do you want to ask the question?
Hello?
Yes.
Can you hear me? Yeah, so what I wanted to ask was, since like we are considering biomedical applications, right? This is like a very are there so based on like how a model predicts for different kinds of different values, right? So like based on what kind of problem we are trying to solve, are there any standards like, okay, if my with how much confidence we can consider that, like, okay, if we are using a deep learning model to solve a particular task or like predict something, like with how much confidence can we accept those prediction results?
Right.
Like, because the degree of sensitivity varies across problem to problem, right? In these biomedical applications. So that's like.
Right. So I think we can ask the same question for computer vision research as well, right? So so in this area, so it's very similar to computer vision, where these so-called standards are generated by a human. Okay. So the level of confidence is really at how these human experts in defining the ground truth of these samples. So yeah, the short answer is Yeah, yes, there is some standard that people follow in defining the ground truth, but the level of confidence is really at the expert that defines that. So the difference between the so-called confidence in this area and the confidence in computer vision is oftentimes in computer vision you can visually see it. In this case, you cannot. So there's a lot of uncertainty involved in that so-called confidence there, but it's very difficult to measure.
Yes. So is it something that like the field, like the entire community is kind of looking forward to also?
Yes, yes. So I'll refer you to this challenge. It's called a dream challenge. So
Okay.
it's exactly spelled as a dream. Okay? It's dream challenge. So yeah, it's a challenge very much like all these Kaggle challenges. I think they work with Kaggle now, so that comes up with different problems that are important to biomedical research and come up with these standards and the ground truths.
Hmm.
Yeah. Mm-hmm.
Okay. That sounds like a very exciting challenge to work on. And thank you so much for sharing your research with us, Dr. Huang, today. And folks can reach out to Dr. Huang offline if there are further questions. Thank you so much for your time, and we'll all see you next week.
Thank you. Thank you, everybody. Thanks, Dorisha. Bye.